we'll get started. So thanks everyone for coming again. Thanks again for coming today to another HPL seminar. First, I'd like to thank Sandro and Biomechanic for providing the coffee and cookies for the rest of the semester. So you can expect this for every week to come. So if that alone entices you to come, that's good. But please come to the science too. Anyway, today we're very lucky. We had a last-minute cancellation, so Dr. Venus Joumaa thankfully was able to jump in and fill the spot, so we can all be here today. So, yes. Venus is an adjunct professor at the University of Calgary. She's been here since, since kind of 2005, with a bunch of breaks kind of in the middle. She's been an assistant professor since 2009 here, or an adjunct assistant professor. She did her undergraduate studies in biochemistry in Lebanon at the Lebanese University, and then she received her master's and PhD degrees in biomechanics and biomedical engineering from the University of Technology of Compiègne in France in 2005. She then came to the Human Performance Lab as a postdoc in May 2005, where she worked until 2011, when she went back to Lebanon to teach, I think. And then she came back in 2013, and she has been with us since, working on projects aimed at understanding the molecular and cellular mechanisms that govern the structural, functional, and metabolic properties of skeletal muscle in health and disease conditions. Aside from doing scientific work, she likes hiking and reading. And her fun fact is that given she studies force depression in single skin and muscle fibers, she also often gets manuscript reviews for 1 or 2 invitations a week to review manuscripts on depression, mental health, and skin. So I guess the algorithms don't work that well. So thank you again for jumping in and helping us, and I will leave this to you now.
Okay. Thanks a lot, Michael. Thank you for the invitation. So I would like to talk to you today about how skeletal muscle produces force. And I'm hoping by the end of this presentation that I would have convinced you that what we know so far about how muscle contracts is not the full story. That there is still lots of research to be done in order to fully understand the muscle in all its conditions. So during this presentation, I will follow this outline. So first, I will remind you of the structure of the skeletal muscle. Then I will talk about our current understanding of how muscle produces passive force, active force, and the force-length relationship. Then I will challenge this current understanding by introducing a property of skeletal muscle, which is residual force enhancement. And then I will talk of some observation that we have done that hints that there is a role of a passive element in active force production. And I will end by conclusions. So let me start with the structure of skeletal muscle. So this is a schematic of a skeletal muscle. So this is the tendon. And this is a cross-section of the muscle made at the mid-belly. So the muscle cell is called fiber. And these fibers are grouped in fascicles. So they are here. These are the fascicles. And so these fascicles are surrounded by connective tissue, and all together they form the fiber. So inside the fascicles, as we said, we have the fiber. So the fiber, like all cells in physiology, they have the usual organelle, like mitochondria, sarcoplasmic reticulum. But what's specific about these cells is that they have what we call the myofibrils, which are the contractile elements of the muscle. So this is a picture of a myofibril under a microscope. So I hope you can recognize the striation patterns, so the dark bands, the light bands, dark bands, light bands, etc. So these striation patterns, because we know that skeletal muscle is a striated muscle along with the cardiac muscle. And in the middle of the light band, we see a line, a dark line, which is the Z disc. So this myofibril is formed by a repetitive structure. It's called sarcomere. And so this repetitive structure is located between one Z line to the next Z line. So what is exactly the structure of the sarcomere at the level of the filaments, at the level of the proteins? So this is a schematic of the sarcomere. So this is the Z line. These are the Z lines again. This is the middle of the sarcomere, the M line. And these are the various filaments. So there are 3 filaments in the sarcomere. 2 are contractile filaments. They are the thick filament shown in green here, and it is in the middle where the dark bands are. And we have the thin filaments shown in blue. And the third filament is Titin, is shown in red. So let's dig into these filaments a little bit more. So the thick filaments are formed of a protein, it's called myosin. So this is a myosin molecule. So this is the myosin tail, and this is the myosin head. And so the myosin head has a spring-like structure here. And the myosin tail forms the backbone of the thick filament. So if I'm talking about the thick filament here, the backbone is the myosin tail. And then the myosin heads are the side pieces that we see, and they are free pieces coming out of the backbone, and we see them here, and they are free to rotate. What about the thin filaments? So the thin filaments are formed of 3 types of proteins. These are the actin proteins, so the actin molecules shown here as spheres in blue. And they are organized, the actin is organized as a strand of double helix. And it has tropomyosin and troponin. These are regulatory proteins sitting on actin. So thin and thick filaments are called the contractile proteins in the muscle. So the third filament in the muscle is Titin. So it's shown here in red, and it is the molecular spring in the muscle. So Titin spans in each half of the sarcomere from the M line to the Z line. And we have 6 Titin molecules per thick filament. So how does this structure produce active and passive force? So let's start with the passive force first. So what is passive force? So passive force is the force produced by the muscle when it is at rest without any activation, without any stimulation, like in the video here. So here we have a myofibril. It's attached on both ends, on this side and on this side. And as you can see, we're stretching it, it elongates. And while it elongates without any activation, so we're stretching, it elongates, and while it elongates, of course, it produces resistance to stretch. So this is what we call passive force. And so the element that produces this passive force is shown in this cartoon here. So you see the sarcomere, when we are stretching the sarcomere, the element that is stretched mostly is Titin. And so the structure responsible for passive force at the level of the sarcomere is Titin. And so it's being stretched. Here I'm showing the passive force in function of the sarcomere length increases. So how is active force produced? So active force, as we all know, needs nervous stimulation and then the release of calcium, and it needs ATP. So how is it produced? So here I have my thin filament again. So thin filament, it is this one. So I have it here again. And so when calcium is released, this calcium binds to troponin. And as you can see, there is a rotation of the tropomyosin, which allows the myosin binding sites on actin, which are the yellow circles here, to be exposed. And so the myosin binding sites are exposed. Imagine they are here. The myosin heads are here, and so they are able to attach. The myosin heads are able to attach to actin and thus produce force. And so this actin-myosin interaction, we call it the cross bridges. And so I'm showing here an example of a cross bridge. So this is the myosin backbone. This is actin. The binding site has been opened, and that's why the myosin head is attached. And this is a spring-like structure of the myosin head. So when there is enough ATP attaches to the myosin head, and the myosin is able to hydrolyze ATP to power its movement. And so it rotates. So it's here, so it's going to rotate, which causes this spring to be stretched. We have a spring, it's being stretched. This means that there is force that is produced. And so this is how active force is produced at the level of the cross bridges in the muscle. And so this is what we call the cross bridge theory. And our current understanding of muscle contraction is based on this cross bridge theory. Okay. So now imagine, so I've shown you one sarcomere. Now imagine that we have so many sarcomeres in the myosin filaments. They have actin, it's exposed, like the binding sites are exposed in front of them. And so they're going to, many cross bridges are going to attach in order to produce active force. And so the higher the number of cross bridges that attach, the higher the force produced by the muscle. And so this relationship between the number of cross bridges attached and the force produced by muscle is like modulated by these two cartoons. So this is, here I have a sarcomere. So again, this is actin, this is myosin in green, and this is titin. And these are here, the lines here, these are the cross bridges. And the myosin here, or the sarcomere here, is at two different sarcomere lengths. And as you can see, when we have sarcomeres at different lengths, the amount of overlap between the actin filament and the myosin filament is different. And so that's why we say the force produced by the muscle depends on the amount of overlap, because depending on the amount of overlap, you will have a different number of cross bridges attached. So I'll let you have a look at these two, these two sarcomere scenarios of sarcomere lengths. So in this case, for example, we have less overlap than this one, and thus the number of cross bridges that could attach are less than here. And therefore, in terms of force, what should happen? So this sarcomere is producing less force than this one. And thus, this is explained by the force length relationship, which was described in detail by Gordon in 1966. So this relationship states that the force produced by the muscle depends on sarcomere length. And this, so we have force here, this is the maximal, the 100% force, and this is a sarcomere length. So this relationship has three regions: ascending limb, plateau, and descending limb. So for the sake of this talk, I'm going to talk only about the descending limb. So as we said before, so the force here decreases as the sarcomere length increases. And as we said, it is because of the amount of overlap that is changing between the sarcomere lengths. So the highest amount of overlap, I'm having the maximum force. As the sarcomere length increases, I'm having less overlap, and so less force. And then in the extreme condition, no overlap, no active force at all. And so our current understanding of muscle contraction is based on these two properties, or on these two theory, or one theory and its derivative, the force length relationship. So to summarize, we believe that the force is produced by cross bridges, actin myosin cross bridges, and the force produced by the muscle depends on the amount of overlap between actin and myosin, and thus depends on the number of cross bridges between actin and myosin. And so for a given amount of overlap, because I will have a given number of cross bridges, we should have one force, one maximal isometric force. So I'm talking maximal isometric force. Okay, I hope this is clear. So this is our current understanding of muscle contraction: cross-bridge theory, force-length relationship. However, I'm going to introduce now a property that will challenge this understanding. It is residual force enhancement. So let me define residual force enhancement. So if we activate a muscle, so all what I'm talking about, I'm explaining it at the level of the sarcomere. Okay, so to not complicate things and things about the muscle, I'm talking filaments, the contractile filaments, and the sarcomere. So if I activate a sarcomere or a muscle, a fiber at a given sarcomere length, according to the force length relationship, so force length relationship, it's going to produce this force. And this is actually happens. It produces about the same amount of force. Now, if I activate the muscle at a shorter sarcomere length, and then I actively stretch it to the same final sarcomere length. So I'm activating the sarcomere, it is here, so it produces this force, and then I'm actively stretching it to get it to this sarcomere. So sarcomere was here, I'm stretching it actively to this sarcomere length. So according to the force length relationship, the force at the end here in both conditions should be the same because the sarcomere length is the same. This is what the force length relationship tells me. However, this is what we get. So this is the activation at SL2, and then the force increases after active stretch, and this is expected. And then it relaxes. Again, this is expected, but it should relax to reach the same level as here. However, it does not. Despite the fact that the sarcomere length at the end is the same, the amount of overlap is the same, the force is higher after active stretch compared to a purely isometric contraction. So this is what we call residual force enhancement. So residual force enhancement, it didn't happen. We didn't see it just in one preparation or once. It's happening systematically. It has been shown in single myofibrils, in single fibers, in isolated muscles, in vivo human muscles. So it has been shown everywhere. Also, the increase in force is not unique. It's not just you increase by this amount and it always happen. We will always obtain the same increase. Depending on how you do the experiments, depending on the amount of stretch that you're doing, the active stretch that you're doing, we could have an infinite number of forces for a given sarcomere length. So we could change the amount of stretch, and so we could get the amplitude of stretch reaching the same final length. We could get an infinite number of forces. Have I intrigued you enough? Yes? Okay, so what's happening then? So what is the mechanism for this? So we want to stay within the context of the cross-bridge theory. So we say this could be because of an increase in the proportion of attached cross bridges after active stretch. So even if we are at the same sarcomere length, maybe after active stretch, we are increasing the number of attached cross bridges. Actually, I have told you before that the force produced by the muscle depends on the amount of overlap and depends on the number of cross bridges. But what I haven't told you yet is that at a given sarcomere length, the cross bridges that are available to attach, so I have cross bridges, they have actin in front of them, the binding site on actin is exposed, but they do not indeed attach. So the number of cross bridges that can attach, they do not really attach. And so, for example, if I have 100 cross bridges that are able to attach, only 20 of them attach. Don't ask me why, please. And so there is a possibility to increase the number of cross bridges that could attach, maybe by active stretch. So active stretch is increasing this number from 20% to 25% or more. Okay, so this is one hypothesis. So how to test this hypothesis? So increase in the proportion of attached cross bridges. There have been some attempts to test this by using mechanical measurements like the stiffness measurements. However, this is a mechanism that's happening at the level of the sarcomere, at the nanostructure. And so the ideal tool to test this would be something that would show us the nanostructure, which is the small angle X-ray diffraction. So what is this? So, you know, when we go at the airport, so we have these X-ray machines. And so they allow people there to see what's inside, to image what's inside our bags. And so here, it's the same principle, but it's obviously a little bit more powerful. So we have this small angle X-ray. It allows us to see what's inside the sarcomere. It allows us to see the cross bridges inside the sarcomere. And so we use this tool. We went to Argonne National Laboratory in the USA, where they have this synchrotron facility, in order to determine the proportion, to investigate the proportion of attached cross bridges after active stretch. And so here, the parameter that is usually measured in these images is not proportion of attached cross bridges, but it is called mass shift, which is an estimation of the proportion of attached cross bridges. And so we did experiments in rabbit psoas bundles. So bundles were isolated from these muscles, then they were skinned and they were mounted between a motor and a force transducer. The experiments were the experiments that I just told you about. It's an activation at a given sarcomere length, and then an active stretch to the same final sarcomere length. And as you can see, we saw, like as usual, residual force enhancement. But what we were interested in was to do X-ray exposure here to determine the proportion of attached cross bridges. And so our hypothesis was that, okay, there is an increase in force, so there should be an increase in the proportion of attached cross bridges after active stretch. Did we find that? Actually, this is the proportion, the mass shift or the proportion of attached cross bridges. So I11, I10, same as the mass shift / proportion of attached cross bridges. So this is the reference contraction and this is the active stretch contraction. And as you can see, there was no difference in proportion of attached cross bridges after active stretch, which means that the proportion of attached cross bridges cannot explain the increase in force after active stretch. So we're back to square one. What is it then? So there has been this hypothesis about an engagement of a passive element. So people thought that, you know what? During passive stretches, so when you stretch a muscle, there is an element that resists stretch passively. So they suggested, why not the same element is also contributing to active stretch? And so this same element would be Titin. So Titin usually resists passive stretch. So people suggested because active here stretch involves muscle stretch, so it could be that the same element that is working passively could also be working actively. And so we did some experiments. We call them proof of concept experiments, just to check whether really a passive element / Titin is involved in active stretch. And so what we did was we investigated the role of Titin by doing a very simple experiment, theoretically speaking, which was degrading Titin, partial degradation of Titin, not total, partial degradation of Titin. And so we did these experiments in single myofibrils isolated from rabbit psoas muscle. So these are the single myofibrils. And so the experiments involved the degradation of Titin, as I said. So we used the trypsin, which is a very powerful enzyme that degrades lots of proteins, but at a very low concentration, it degrades Titin only because Titin is very low, so it is very susceptible to degradation. And so we used the trypsin, we incubated the myofibrils in trypsin to partially degrade Titin, and we saw that Titin was degraded by about 50%, while all the other proteins were left intact. And then we did the experiment same as before. So we did activation, active stretch when Titin is intact, activation, active stretch when Titin is partially degraded. And so our hypothesis was that there should be, so if Titin is really involved in the increase in force after active stretch, when Titin is partially degraded, there should be a reduction in the force after active stretch compared to when Titin is intact. And so what did we find? So this is the stress in the myofibrils in function of time. So this is the activation and this is the active stretch. And so as you can see, the force, so here we didn't perform a purely isometric contraction because a myofibril is very sensitive. It does not take many activations. So we're comparing to force before stretch. And so as you can see, the force increases by 50% compared to the force by 46%. So this is the force increase. It increases by 46% compared to the force before stretch. So what happens so this is an intact Titin. What happens when Titin is partially degraded? So our hypothesis is that there should be, instead of 46%, we should have, because Titin was degraded by half, we should have about half increase in force instead of this 46%. And this is what we found. So this is the activation when Titin was partially degraded, active stretch, and this is the force after active stretch. So as you can see, there is a decrease in the amount of force after active stretch compared to this one, which is 28%, like the increase here compared to here is 28 compared to 46, which like we were happy when we saw that, because it confirms the hypothesis that Titin is involved in the force after active stretch. However, I'm not sure if you have noticed, we did not expect this. So when we activated the myofibril, they produced less force than the purely isometric contraction, less than when Titin was intact. So there was also a decrease in the purely isometric force. When you activate the muscle, just when Titin is partially degraded, there was a decrease in this force, which was not expected. And so we could say that we're happy. It seems that we have shown that Titin really, which is a passive element, is contributing to active force production after active stretch. But we were surprised to see that it also, surprised but happy to see that it also contributes to force, to the isometric force. But now, all of these are observations. We need to think about a mechanism. So what's happening? How is Titin able to modulate active force? So we looked into the literature, and it seems that something was proposed by Cazorla in 2001. So this is our half sarcomere. So this is the half sarcomere. This is the M line in the middle. This is the Z line, and this is Titin. And so these people suggested that Titin, as we said, is a passive element. So it produces force when it's stretched. So this is the vector of the force produced by Titin. But then this vector could be divided into a radial component and a longitudinal component. And so this radial component, imagine the effect of this force on the sarcomere, it would be to bring the filament closer together. So it would be a compression of the filaments together. And so Titin, so as we said before, just to remind you, I have cross bridges, they have actin in front of them. Now I'm bringing them even closer to actin. And so this means that they could attach easily to actin. And so this is what we mean by modulating the lattice spacing, bringing the filament closer together. So it could be that Titin is modulating the isometric force here and the active force here by compressing the filament, bringing the filaments closer together. So how to test this? So this is the aim of our next experiments, was to investigate whether Titin regulates the contractile filament by compressing the contractile properties, by compressing the filaments. And so we did the experiments in single skinned fibers. So instead of the myofibrils, we did the skinned fibers, single fibers, because they can take more contraction, many contractions. So this is a fiber. It's attached between again, force transducer here and a length controller. So this is the fiber here. And so how did we test for our for the purpose? So what we did experiments in intact fibers. So Titin is intact. We did the active stretch experiment. Then we did Titin partial degradation. So Titin partial degradation, as expected from before, would reduce active force before an active stretch. Now, in order to test whether Titin is reducing force by compressing the filaments, what we did was, in the Titin partially degraded fibers, we added a chemical, which is dextran. It compresses the filament. So if Titin was doing its job was increasing the force by compressing the filament, when I have Titin partially degraded, and I have this chemical that compresses the filament, I should have the same result as in intact fibers. And so this was our hypothesis. So hypothesis was, if Titin is acting by compressing the lattice spacing, when Titin is absent, is degraded, but I have dextran that compresses the filament, the spacing between the filament, I will have a recovery of the force. What did we find? So this is the stress, just for your information. So here to show you what we're doing. So this is the stress, this is the activation, and this is the active stretch in intact Titin. So we're repeating the experiments, but Titin is degraded next, and then Titin degraded plus dextran. And so the parameter that we looked at here is a PCA 50, but please treat this parameter as similar to force. So imagine it's just force. So when Titin is intact, as we saw, so this is the reference force, and this is the force after active stretch. It's higher than the reference force. This is expected. When Titin is partially degraded, there is a reduction in the reference and a reduction in the force after active stretch. We saw this before. Now, the next result is about dextran. So if you put dextran, are we going to recover these values to here? If this happens, this means that Titin is playing its role by compressing the filaments. This is what we got. So the reference force is recovered. So to the values observed in the Titin intact conditions. But the force after active stretch is not recovered. This complicates things. So what does this mean? So this means that likely Titin is acting on isometric force here when it's here, like it's acting on isometric here force by compressing the filaments. But when there is an active stretch, compression of the filament is not enough to increase the force by the amount observed in residual force enhancement. There must be another mechanism that's acting on the filament in addition to the compression of the filaments. And so what is this mechanism? So this mechanism was suggested by Leonard et al. In 2010. And so what did they say? So they say that this is the half sarcomere again. So this is in the purely isometric contraction. So the length of Titin here is the full length. You activate the myofibril, you have this isometric force. Now, if you activate the fiber and actively stretch it, so this is what happens. During the first activation here, in the presence of calcium, Titin binds to actin, which causes its length to be shorter, just to have a shorter free length Titin. When you're stretching to the same final sarcomere length as in here, so we're stretching the sarcomere to the same final sarcomere length, we are stretching in the active stretch condition, we're stretching a shorter spring compared to when you're stretching a long spring. Of course, when we're stretching a short spring, the force is going to be higher. And that's why the force is higher after active stretch. So the only difference is that in purely isometric contraction, we're activating the sarcomere when Titin is at its full length. In here, the Titin somehow got shorter. And so when you're stretching it, it produces high amount of force. And that's why we see residual force enhancement. So this mechanism needs lots of testing and lots of work to be proven correct. And so the first step in it is to do some labeling of Titin So labeling using antibodies. So we will have antibodies that are fluorescent and they are located, they would attach titin at different regions. And so we will have a myofibril like this with titin labeled, and then we stretch and we see whether which segments of titin are being stretched. Is it the whole segment that's being stretched, or like in this case, or we have like small segments are being stretched. Okay, so these experiments have been done. So some preliminary experiments have been done, but the results are still not conclusive. But more experiments are in progress in the lab. So I hope I have convinced you. Maybe you would put some question marks on our current understanding of muscle contraction by saying that there are some properties that are not being able to be are not explained by our current understanding of muscle contraction. And hopefully, so the aim of all of this is to be able for you to apply this to other research and to other concepts. Like, do not take everything as, okay, this is proven, this is the truth, we need just to study it and to accept it. Always put question marks on what you are studying and look for, okay, does this really make sense? And so always ask, is this the whole story? And so we have provided evidence that there is an engagement of a passive element during muscle activation. And so a passive element should be considered when we are thinking, we are explaining after active force production in various conditions. The mechanism, we don't know yet. Lots of research is still to be done. So it could be binding to actin, it could be somewhere filaments working on the compression of the filament. It could be compression of the filament and change of the conformation of the cross bridges, change of the amount of the stretch in the spring. Its mechanism is open, but at least there is some evidence that we need to look further into the mechanism of muscle contraction. And at the end, I would like to acknowledge the people who contributed to the X-ray diffraction experiments, the people who contribute to these experiments, the Herzog group, and of course, I need to thank Walter for having me in his lab for all this time and allowing me to do all of these experiments. So with the sky is the limit to my dreams in terms of experiments with Walter. So, and also thanks to the funding agencies, and thank you very much for your attention and for bearing me talking about very basic mechanical properties of skeletal muscle. Thank you.
Right. So now we have time for questions. So if anyone
I'm curious about the study you published in 2020. I'm wondering, could you not even expect the opposite finding in basically reduction in cross bridges due to the stretch that would maybe mechanically actually break the cross bridges?
Okay. Yeah, it's possible, but in terms of the cross-bridge theory, like we have a given sarcomere length and we have an increase in force. So the logical explanation would be having more cross bridges. But of course, there is a possibility for less cross bridges and then another element taken over. So that's a hypothesis that could have been suggested as well. Absolutely. And that's a very interesting comment, actually, because as you can see from the results, there is a not significant, but there is a trend towards a decrease in the proportion of attached cross bridges, which could agree with what you're saying.
I noticed in some of the cartoons that you have, it looks to me like the Titan crosses the set line and even and
Mhm.
the M lines. And in some maybe it didn't so much, but I guess the question is, does Titan, is there just, does it just go from one Z line to the other Z line, or does it go from the Z line to the M line, or does it cross sarcomeres?
Actually, very good comment as well. So at the Z line, yes, Titan, it crosses to the next one on the other side. And it crosses just by ends, like end of Titan from here and end of Titan from the other sarcomere. And there are lots of proteins that are making the connections as well. And we believe that this is this is very important because it causes some crosstalks between sarcomeres, like sensing of the mechanical properties between sarcomeres. And there are many studies that are focused on how this interaction is happening at the level of the Z disk between one sarcomere here, one Titan on this side, and one Titan on the next side.
And the belief is that it's more sort of it's sensing related rather than some mechanical interaction between sarcomeres. Is it one and the same?
Sensing, mechanical sensing,
Yeah.
sensing of force, sensing of change in stiffness.
Okay. I think we talk about this every time somebody brings up residual force enhancement or residual force suppression. It's kind of fun to think about. Like in your mind, is there a functional benefit to this phenomenon?
Okay.
Yeah.
So that's a hard question for me. Working at the cross bridge level. Okay, so usually in everyday movement, we do eccentric contractions and we are stretching our muscles. However, reaching a steady state doesn't happen often. So the steady state residual force enhancement may not contribute to our everyday movement. However, having Titin is playing a role in the muscle and regulating, because what we're saying here is that it regulates active force production. So we believe that we do not need to wait until we reach a steady state for Titin to get involved in the regulation of active force. And so it could happen in purely isometric contractions. It could happen just at the top, at the end of the stretch, not necessarily waiting until steady state.
So it helps.
Yes.
Because Venus did one study and where she showed that, where she also measured the energetics, and she showed when you were in the enhanced state compared to the isometric reference state, so after stretch, the ATP required, so metabolic energy required for this higher force in the enhanced state was actually less than the lower force in the isometric state. And therefore, you know, I was just asked the same question in Omaha last week. And so sometimes my answer is that, you know, when we walk around and we run around, and so, you know, you only use 10%, 20% of the force. So getting a little bit more force, maybe you don't really need the force enhancement for that. But maybe from an evolutionary point of view, the fact that you save energy through the action, metabolic energy, through the action of Titin might actually be more important than the fact that you get more force. That you get more force or the same amount of force for much less energy. And maybe that's the advantage, that you save 20% of energy whenever you do a stretch shortening cycle and you walk around, you go up a mountain or down a mountain. So maybe it's an energetic thing rather than a force thing that is actually important in everyday life.
And is it the opposite in that it's less energetically efficient for force depression?
Actually, no. In force depression, we saw that there is a reduction. So when the force is decreased, there is a proportional decrease in ATP consumption.
Okay. So you only see this efficiency difference when you do
force enhancement. Exactly. Yeah.
Before we stop talking. So okay, now leaving force enhancement, force depression aside, this observation that Titin plays a role in just isometric contraction. have you thought about functionally why that would be?
Yeah.
Do you think of that in terms of it? Maybe it has something to do with energetics, or do you think it has more to do? I don't know.
Yeah. So, you know, the way we think Titan might affect the cross bridges and isometric force is by changing the conformation of cross bridges. So I said that you have rotation of the cross bridge and stretching of the spring. So the amount of stretching of the spring could be different depending. So this is what we mean by conformation. So if the conformation of the cross bridge is like this, the amount of stretching is bigger than when I have it just a little bit here. So Titan could modulate the conformation of the cross bridges. And thus, yes, we know that one cross bridge to cycle, it uses 1 ATP. So if it uses the same 1 ATP to produce more force by having its spring stretched more, this is beneficial.
Thanks, man. That was a really interesting talk. I have a question. Does sarcomere length change between fiber types? So between type 1 fibers, 2A fibers, and 2X fibers, does sarcomere length change? That's part one of my question.
Okay. I don't fully understand. So, of course, sarcomere length changes in various conditions when you stretch the muscle, if it's fast or slow. But do you mean if it's in a mixed muscle?
Well,
so you have slow fibers and fast fibers, would they have different sarcomere length?
Exactly, yeah.
I don't know.
Okay, cool.
That's a very good question.
No,
I don't know.
Yeah, and that kind of goes on to my next question of what Walter kind of mentioned with the ATP consumption, because obviously 2X fibers, they consume a lot more or use a lot more ATP, but with their isometric contraction, they can go a lot quicker and a lot faster, but they're not as resistant to fatigue as maybe a type 1 fiber. So with your enhancement experiments, I was just going to ask, is there a difference between fiber types in terms of the type of enhancement that you do get? Because maybe type 1 fibers are more resistant to fatigue, are they able to stay in kind of conform more contraction, more of that type?
Yeah.
Just wondering about that.
You know, yeah, again, very good question. So there is this study that was done by Linke student, Prado, and they showed that there is no correlation between Titin isoform. So what you're saying, I think about it as Titin isoform, because when I talk about role of Titin in force production, I think about the different lengths of Titin and its force. And so what they found was that there is no clear correlation between the fiber type and the size of the Titin, which is surprising. Because most like slow fibers, I feel like I've seen like they have long Titin, while the fast ones, they have shorter Titin. But they did lots of muscle in rabbits, and then they did not find a correlation between the Titin isoform and the type of fiber. However, testing force enhancement in different fiber types, this is a project that is on our minds, it has been on our minds for some time. We haven't done it, but it should be done.
That was great, as always. Really interesting to listen to all this about sort of cutting edge science. I feel like, you know, especially our understandings of the theory of muscle contraction. But is there any, kind of going back to what Brent was asking about, are there any physiological conditions or illness states where it's Titin is known to be impaired, or you know, you might be able to observe something functional in the state that you basically created with the experiments?
Yeah. So there were those experiments performed by Jeff Bauer, and he tests force enhancement in elderly people. And surprisingly, isometric force was reduced in this population, while residual force enhancement was not. When you actively stretch the muscle and get Titin involved, residual force enhancement was not reduced, like relatively speaking. Of course, the amount of increase relative to the isometric force was not reduced, which means that Titin has a functional role, especially when the muscle is damaged, when there is something happening to maybe active force, it could play a role. But Again, these are just observations, one study here and there, but they have not been systematically studied.
Is there, I mean, I was reading recently that Titin could be quite a, like a maybe regulatory in terms of how muscles regenerate hypertrophic response.
Absolutely.
But is there any way that you could take your model that you do with with a myofibril and sort of like take an animal model to be able to sort of degrade Titin on some level just in the muscles to see functionally how that changes, or would that just have too much systemic harm?
Exactly. This has been tried, like people have tried to degrade Titin and the mice are not viable after that, so they die. However, there are some Titin knockout models where Titin pieces here and there are being eliminated, deleted, having Titin longer, having Titin shorter, and these experiments have been done. And of course, when Titin small, like we have a model in the lab that we have used as the MDM model, which is like a small change in the number, in the amino acids, like it's a very small chain in the Titin, which results in the mice being like they would live just weeks, 4 or 5 weeks, and then they die. And their force is compromised. It's interesting. Just a small change in Titin results in a huge functional damage, which of course supports the idea that Titin is not just this passive element that if we think about where our muscles function in vivo, you think it never works because you need to stretch the muscle a lot to have Titin stretched. And so it must have another role in the muscle. Otherwise, why would the cell put lots of energy in producing this huge protein without using it?
Few questions about that experiment you did. So about degrading Titin, is that at the or something chemical you use, it will only degrade Titin, but you will other things. But how can you actually see that? Can you measure something to verify that?
Yeah. Yes, very good question. The active force was reduced after Titin degradation, partial degradation, which means that the first thing that comes to mind is, okay, are we degrading the actin and myosin as well? And so we did experiments like the spectral mass spectrometry, which tests the proteins that are in the muscle and in Titin intact and in Titin after degradation. And we saw that there was no change of other proteins except Titin.
But when you say there is no change in other proteins, does it mean that it is just there, or does it mean that it is actually functionally intact?
Good point. It's there. We do not know if it's functionally there. Yes, good point.
I imagine if you totally destroy the Titin, maybe even though everything else is intact, maybe it's no longer.
Exactly. So we have tried 100% Titin degradation and the active force is completely gone. And we do not believe it's because Titin contributes to active or produces active force that much, but because the whole structure is gone, is not functional anymore. Yes. But yes, very good question. Yeah, in terms of the trypsin.
It's just a quick comment, it's not that the whole protein Titin is destroyed and everything else is intact. But Titin has this one weak segment, and so you really cleave. It's kind of like going in with a scissor and cleave it at one point, and then it becomes useless because it's not connected anymore between the myosin and the Z line. So it's not that the whole Titin is digested away and it's gone, and everything else is in contact. You just really clips it at one teeny tiny point, and then it becomes functional.
Well, I don't have any question, but I got a question from the audience online. So there you go. He will ask you the question. Go ahead there.
I just had a quick question. So do you happen to know how stiff the actin and myosin proteins are, like when they're in their attached cross-bridge conformation? Like, could it be possible that one of them gets pulled slightly when you do the active stretches? because they're in that conformation and then you're stretching it. So it might like shift it a little bit, or they might get pulled if they're a little bit slack. Do you just happen to know how stiff the proteins are themselves?
Yeah. So this happens during the transient phase. So I think I have just one. I'm not sure if you get let's see the figures. So we believe that during the stretch itself, the cross bridges are being stretched, and that's why the force increases here. But once we reach the steady state, everything has returned to normal. And so the cross bridges, they should go back to their original cycling states. So yes, you're right, at the transient phase, but then at steady state, it's purely isometric contraction. There is no change in the, like, supposedly no change in the cross bridge cycle. It goes back to normal.
Okay, yeah, that makes sense. Thank you.
Thanks, Daria.
So I'm wondering about muscle spindles. So muscle spindles also have contractile elements. And I'm actually wondering whether those are the same contractile elements we have in this regular muscle. And then if so, basically, then that also means that the same rules that we currently think of, that they also apply to muscle spindles.
Okay, so I do not know the exact structure in muscle spindles, but I know like the fiber, even the fiber types there, it might be specific to this region of the muscle, but I cannot answer that.
Well, I got another question from Robert Griffith. Go ahead, Robert.
Hi, Venus. I enjoyed the talk. Thank you. I remember decades ago when I used to look at the actin myosin filament plan for muscle. I used to think that if the sarcomere was pulled to long length, all the myosin filaments would drop out into a little pile of proteins, and therefore I thought there has to be something connecting the myosin to the Z line, and the Titin was the answer for that. But I also, in looking at the same sketch of actin and myosin in the sarcomere, you see the actin filaments always drawn as these lovely straight blue lines. You've got them up on the screen right now. And I'm wondering exactly the same argument. Why would the actin filaments be these lovely straight lines poking out into thin air, well, into nothingness and aligning, unless they're running inside little microtubules that keep them aligned? Or has anybody actively looked to see if there's a connection between the end of one actin filament and the next actin filament, in the same way that Titin joins the myosin to the Z line? So I guess it would act as a parallel elastic element, but it would also, as I could see the argument for the actin filaments to point towards each other and not collapse. Maybe they need to have something joining them. Has anyone actively looked to see it for a protein joining the two actin filaments?
I don't believe somebody has looked at proteins joining the filaments, but what I know is that actin is not like sitting in the air alone like this. It's sitting on a protein, another protein. It's called nebulin, and it's working like a scaffold for actin. So it's holding it in space somehow, as far as I know.
A bit like a microtubule to run down, or
exactly.
Often it's actin within ordinary cells, non-muscular cells. Actin provides the structure, the internal structure of ordinary cells. So I could see, actin filaments are known to provide the structure inside a conventional cell, but you're saying there's another thing that provides a structure even for the actin to sit on.
I believe so. Yeah, it is a protein called nebulin.
Oh, yes. Yeah. And you don't think it contributes to the behavior of the sarcomere?
it has been neglected so far. I know that Hank Granzier and Kohn Ottenheim, they've done lots of work on nebulae knockout mouse, and then they found that it also like causes a big problems in the muscle. So, but I do not, I cannot say I'm expert there, but it's more physiological role, sarcomerogenesis rather than terms of mechanical, direct mechanical role for nebulae. But everything is open to discussion, absolutely. As long as it
Thanks,
has
Vince.
not been directly tested, we cannot say that it cannot happen.
Exactly. Yep. Thank you, Vince.
Thank you.
Questions? Walter?
The, you also find it interesting the Cazorla study there, because we draw the sarcomere like that, but in reality it's not. If you have the actin and myosin very overlapping, then you are at 4 microns, and the myosin is 1.6 microns. So that's about 2.4 divided by just about 1.2 microns. So you have 1200 nanometers from the end of the myosin to the Z line, and from the actin to the myosin to about 20 nanometers. We have like 20 over 1200. So that angle that we show in these things is always way too steep. It's really essentially going parallel. I mean, it goes 20 units in that much sarcomere, it will go 20 units in one direction, like it goes 1200 units in the other direction. And therefore, that whole compression thing, I never really thought very much into it. So, would you
I agree.
agree? Or,
Yeah, totally. I agree. Yes. So I think our compression, 4% exon, would be more compression than what Titin really does to compress to get the filaments closer together. Absolutely. Yeah.
the other thing, of course, that we talked about, and just throwing it out there for everybody to think about, you can see Titin in the synchrotron. You can see it in electron microscopy. So what experiment can we ever do to see that Titin actually binds to actin?
Yeah. So I'm hoping that Armand will give us some answers soon with his labeling experiments.
Yeah, so the Mike Duval, of course, showed a
And-
little bit that the proximal segment there, is not elongating anymore in the active state.
Yeah.
And so we interpret that it binds to actin, but maybe something completely different happens. Maybe it binds to something else. Maybe when it's activated, that's different things. So the experiment by Mike and what Armand is trying to do right now are really very good. And if you repeat that and have the full force, it would be really nice to see that proximal segment is not elevated. But whether or not it binds to actin, we actually still, at least I would think we still don't know it. I mean, there would be good evidence, but how do you show that it actually? How do you show that it binds? How do you show what binds with what? And how do you show what configuration needs to exist that the binding happens, and why does it not happen when it's passive?
Yeah. So the binding itself has been shown in vitro. Like they have isolated Titin, isolated actin, and put them in the what they call sedimentation, co-sedimentation experiments, and they saw that they bind and there is interaction between them. But to translate this into a physiological role, there is nothing.
Showing the motility assays is that when you add Titin, then the actin film is slowed down, which then would suggest, oh yeah, maybe it binds a little bit, but the force cannot be very big. Because otherwise it
detaches.
Because it doesn't stop sliding, it still slides, and it's also in a structural arrangement, motility assay. It's in a very different arrangement, and I always think that having the structural arrangement of the sarcomere might really be important, rather than having a kind of a lawn full of myosin sticking up, and then the actin crawling on it. And then you show Titin in it, and then it slows it down a bit. I'm not, I think those are beautiful experiments, but I'm actually not sure what they really mean, except that we might have a very weak transient binding, but we know already that Titin binds to actin at the very, very end of that, you know, the last 50 nanometers, very strongly. But so we know that, and you know, maybe that's just located gas, and maybe that's just a reflection of that, and have nothing to do with some additional binding that might be happening in the active state. So
Totally.
so Brent has the idea now.
No, I wanted one of you to summarize Mike Duvall's findings. Like, why wasn't the labeling good enough to sort of
What Mike essentially showed is that when you pull the myofibril sarcomere passively, then you get an elongation of the entire Titin, you know, from the Z line there to the myosin. When you activate it, then what is shown in B there, part of that, you know, that what we call the proximal segment, the one close to the Z line, is not elongating, but the rest is elongating. It takes up the entire half sarcomere elongation. So we know something's happening there. Titin is not elongating, but you know, and a lot of people think, okay, that's absolute foolproof, yeah. And there's several people out there that take this for granted now. Yo, Titin binds to actin, no questions asked. But I'd actually like to see it. You know, I'd like to see it. I'd like to know that it actually does that. But you can't see it in synchrotron. You can't see in electron microscopy. You can't see that.
How does it unbind?
Same thing with the question. So we know that when we deactivate the muscle here, the force is still higher after deactivation of so it hasn't unbind yet. So it takes time. And I know that Shuye has done experiments showing that in single fibers, it never unbinds, it never detaches. Like she waits and waits and waits and do different various protocols like shortening and waiting and then shortening again. And it seems that it does not unbind in vitro. I don't know in vivo what happens.
In vitro, in the skin fiber.
In the skin fiber.
Entire muscle, it seems to
unbind.
Yeah,
Exactly. It unbinds.
Would it take energy to unbind?
Some enzymes, some proteins that are not in vitro, but they are here.
You have a question?
Yeah.
Why don't you go there?
Sure. All right. So the one thing I wonder is, like, why doesn't just lengthening the muscle bring the cross bridges closer together? Like, if you integrate the tight, you still lengthen everything. If it's constant volume, everything should come close together. But is it that because there's no tight dimension of stability in the structure?
Exactly. So in skin fibers, this concept of constant volume is not respected. So yeah, we may not have compression of the filaments.
And then at the whole muscle level, how much passive stiffness is tight responsible for? Is it majority still?
At the level of the fiber?
At the whole muscle level.
Oh, whole muscle level. So I believe originally it's the collagen, and then as you stretch, there is contribution of Titin. In recent experiments, Hank Granzier, he showed that Titin contributes a big amount to the passive force at the whole muscle level. But in terms of, again, the sarcomerogenesis, like if Titin is not there, the passive force is not there, the structure of the sarcomere is not nice. I know that I went out of the question. I'm not directly answering the question because the exact numbers, I do not know them. How much initially and how much afterwards, I don't know. But I believe initially it's collagen.
Is there any muscle that doesn't have tight across all muscles that have been studied? Is there always tight?
Yes, there is always Titin across all skeletal muscle, cardiac muscle, even in smooth muscle, there is a form of Titin as well.
So I could just add, Joumaa is going to be trying to do some experiments where the Titin analog exists. They're not really Titin. So So for example, in the software, the fruit fly, there's this Titin analog, it's called projection, but it's only about 1 megadol, 800 kilodaltons 1 megadol, so not 3.4. So it's much, much shorter. And so we think of it as an evolutionary precursor of Titans. Then in certain crab muscles, there is also another protein that's kind of like structure like a Titan, but much, much shorter. And it's really interesting because some of you might know that crab has this ability where it can exert an enormous amount of force for a very long period of time where it stays closed. That is because of an engagement of that Titan analog that then the crab muscle exerts an enormous amount of force. There's no energy cost. So the idea is, we want to really see how that functions as an evolutionary, precursor. And we know that this precursor can attach between active myosin and can exert a lot of force. But there's a regulation how it attaches and detaches. And so maybe modern skeletal muscle has that mechanism, where it can transiently attach and detach and get that mechanism. So by studying by some of these muscles that don't really have a Titin or a Titin analog, I hope to maybe learn a little more about that in a few years. Oh no, she's 6 today. Yes, she called in. So she will try and do that. Yes.
I guess I could see that in like muscles that you don't want a lot of excursion in. You want a lot of fighting, I guess. You want purely isometric contraction.
And that's philosophy of it, because you can only stretch the muscle at 5 or 70%. So it's very, very short and it gets incredibly stiff. But of course, it uses the muscle differently because you activate the muscle once and then your wing flaps back and forth 3-4 times, and then you activate it again. So the muscle is not activated every time it shortens. It's only activated every 3rd or 4th or 5th time, and then it flaps elastically back and forth. So you want to have an elastic structure that you kind of tip and then it goes back and forth, and then you hit it again and goes back and forth. So it's a very different muscle. But does that muscle have force enhancement when you stretch it? First question, very simple.
So we know that in cardiac, it's a cardiac muscle. It has very short Titin. It's very stiff, and we have shown force enhancement in there as well.
We have shown force enhancement. 2 other groups, one in Germany and one in Canada, have shown there's no force enhancement in cardiac muscle, and I have no clue how they got to that stage. In fact, the German group has shown that when you stretch a muscle, not only is there no force enhancement, there's no increase in force during stretch. And everything we have stretched, cardiac, skeletal, when you stretch a muscle, force increases. So I have no clue. And it's published in good journals, it's cited like crazy. And Venus and other people need to do something about that. Because I think it's just really the wrong information out. But the McGill group, we know why they didn't see the force enhancement. It's a slippage thing that they did. That of course doesn't work. But the German group, I have no idea that they didn't find any force enhancement. Cardiac muscle, but it's one of those fun feeds, where 2 groups do the same kind of testing. One group finds it, the other one doesn't.
Go figure.
But Venus and Shuye and so on, I have a lot of testing.
Exactly, that's my results.
It's really interesting because Venus published that result in an abstract, I don't know, some 10 or 15 years ago by physical science. But okay, you don't need to write a paper. This is clear, of course it has force now, nothing new. And then there's one paper after the other 5 years later that come out, oh, there's no force now. Oh, great news. What is happening here now? And you're going, what are they? Of course, we believe our results better than anybody else.
Absolutely.
Any other questions?
I was just wondering what the mechanism is for the DEX and how does it compress the surface?
I think it's by osmotic pressure. So it's a kind of a gel outside the fiber. So it absorbs the water from the fiber, and so it compresses it. I believe so. But 4%, yeah, it's I don't know, it might have compressed more than what Titin does, as Walter suggested.
So if there are no other questions, let's thank our speaker once again.